السلام عليكم أعزائي الطلاب. This is the third lecture regarding amino acid metabolism. The objectives of this lecture include discussing the reactions of urea cycle, a few notes about catabolism of carbon skeleton of amino acid, and a few notes about creatine and creatinine metabolism. We knew from the previous lectures that the amino group of amino acids by a combination work of transamination and oxidative deamination will be removed as ammonia. Also, ammonia can be produced from other metabolic routes such as non-oxidative deamination and other routes. This ammonia is relatively toxic. It inhibits hematopoiesis, that to say it inhibits red blood cell formation inside the bone marrow, so it may cause anemia, and it can acquire a proton, converting it into ammonium ion. Ammonium ion have a charge, so it disturbs the acid-base balance. In comparison, we have urea. So what is urea? What is urea? Urea is a diamide of carbonic acid. We can see that urea contains two amino groups. This amino group, amide group, and this is another amide group. So by formation of urea, we can get rid of two potentially harmful ammonia. Urea is solely produced in the liver, so it is only safe to produce ammonia inside the liver. We can see that urea have no charge. So unlike ammonium ion, urea is not charged, and that is why it is safe. It does not affect the acid-base balance. Only in very high concentration, urea may affect or inhibit the bone marrow and cause bad effect. This is a useful link, a simple YouTube video about urea cycle. You can see it if you like. So why urea cycle? Why we have cycle? Urea cycle, as you can see, ornithine enter inside the mitochondria in exchange of citrulline, where it's combined with carboxyl phosphate to form citrulline. Citrulline then combined with aspartate by argininosuccinate synthase, forming arginosuccinate. Then by lyase, it will form arginine, and arginine will lose its urea part, this is the terminal part, to form ornithine again. Urea then can be excreted by hepatocyte and then excreted by the kidney in urine. Now let's speak about how many ATP is required in this cycle. First, we have 2 ATP required for formation of carboxyl phosphate. So 2 ATP is required. 2 ATP is converted into 2 ADP. Inside the mitochondria, carboxyl phosphate is formed. Then argininosuccinate synthase required 1 ATP. This ATP is converted not into ADP but into AMP, adenosine monophosphate. So we lost here 2 ATP, and here we lost 1 ATP. But this ATP is converted into AMP, adenosine monophosphate. And there is a reaction involve a combination of AMP with ATP to form 2 molecules of ADP. So we can say that in this cycle, in fact, we lost 2 ATP. We lost 1 ATP by converting it into AMP, and this AMP take another ATP molecule to form 2 molecules of ADP. And so we in fact lost 4 ATP. So in sum, we can say that urea cycle requires 4 molecules of ATP. This is it. This is an acetyl glutamate. An acetyl glutamate formed by combination of acetyl-CoA with a glutamate. So taking a high protein diet will produce the substrate and the upregulator of carboxyl phosphate synthase enzyme. there will be increase in action of this enzyme, there will be increase in carbamoyl phosphate synthesis, and there will be increase finally in urea production. A bit of diet of an acetyl within the scope of amino acid metabolism. Now we get rid of the amino group of the amino acid. The carbon skeleton is exposed to the catalytic pathway. It can be hydrolyzed or degraded to produce ammonia or various other intermediates. Eventually, all the hydrocarbon skeletons of amino acid converted into acetyl-CoA, acetoacetate, pyruvate, or other intermediate of acetic acid. This cycle represents, we can see that some of the amino acids such as alanine is converted, of course, alanine is converted into cysteine, glycine, hydroxyproline, serine, threonine. All of these amino acids can produce pyruvate. Isoleucine, leucine, tryptophan can produce acetyl-CoA. It is not alanine can be converted into pyruvate by transamination, so that is why it is easy to remember alanine will produce pyruvate. Also, for example, glycine can be converted into serine, and serine can be converted into pyruvate. So it is not very difficult. For example, histidine converted into glutamate, and the glutamate by transamination converted into alpha-ketoglutarate, also glutamine. But if it is lost its amino group, it's converted into glutamate. The glutamate is converted into alpha-ketoglutarate. Tyrosine, it have a hydroxyl group. If it lost it, to convert it into phenylalanine, and phenylalanine enters acetic acid cycle as a fumarate. Asparagine lost its amino group, converted into aspartate. Aspartate by transamination converted into oxaloacetate. So generally we can divide these amino groups, amino acid, into some which are produced intermediate of acetic acid cycle, such as arginine, glutamine, proline, glutamate, others, and others which produce acetoacetyl-CoA or acetyl-CoA directly. Since both acetyl-CoA and acetoacetyl-CoA have no important or have no existing method or route to be converted into glucose, these amino acids are called ketogenic amino acid, because the resulted acetoacetyl-CoA only can synthesize fatty acid, while those who produce alpha-ketoglutarate, succinyl-CoA, fumarate, or oxaloacetate are called glucogenic amino acids. So we can see, for example, isoleucine. Isoleucine can produce succinyl-CoA, and also it can produce acetyl-CoA. So they are called both glucogenic and ketogenic amino. Leucine and lysine. Leucine and lysine have no way to be converted into one of the intermediate of acetic acid cycle. So we cannot by gluconeogenesis produce glucose from the carbon skeleton of leucine and lysine. They are strictly ketogenic. Isoleucine, we can see it can be glucogenic and ketogenic. It can provide succinyl-CoA or acetyl-CoA, and the aromatic phenylalanine, tryptophan, and tyrosine, these also produce either intermediate of acetic acid cycle or acetoacetyl-CoA. So they are both glucogenic and ketogenic. Glucogenic. Alanine, arginine, asparagine, and the others, all of them are called glucogenic. So we have glucogenic, ketogenic, and glucogenic and ketogenic amino acid. Remember these: leucine and lysine as ketogenic, isoleucine, phenylalanine, tryptophan, tyrosine as glucogenic and ketogenic, and the remaining amino acid will be the glucogenic amino acid. One may ask, since those glucogenic, for example alanine can produce pyruvate. Also, pyruvate also can be converted into acetyl-CoA, and acetyl-CoA can synthesize fatty acid. So why we don't call them glucogenic and ketogenic? We'll discuss this lately, not in this lecture, but in discussion in the group. Reach the end of the catabolism of amino acids, carbon skeleton, and the amino group of amino acids. Now, few notes about creatine metabolism. Creatine is a very important compound. It is vital for muscle metabolism because it is a very high energy store, and it can produce energy rapidly in the myocytes. For the synthesis of creatine occurs inside in the liver by reaction of arginine and glycine by enzyme called transaminase, result in the formation of ornithine. Ornithine, this is the intermediate of acetic acid cycle, and guanidinoacetic acid. This reaction occurs in the kidney, liver, and pancreas, most importantly inside the liver, not in the muscle. Remember this. So creatine synthesis is not inside the muscle, it is mainly in the liver, also in the kidney and the pancreas. Now, guanidinoacetic acid will acquire a methyl group from S-adenosyl methionine. S-adenosyl methionine comes from a reaction between methionine, the amino acid, and ATP, where the adenosyl triphosphate loses its phosphate and attach to the methionine, forming S-adenosyl methionine, which is a very important methyl group donor. It will help in transmethylation reaction, various transmethylation reaction. We'll see this in the structure in the following slide. Transmethylase will result in the formation of creatine and S-adenosyl hormone reactions, and then creatine will act with ATP by the enzyme creatine kinase, CK or CBK as we called it, result in the phosphocreatine formation. This is the very high energy compound. It have a negative 9 kilocalorie of energy. It is similar to that of ATP, and it serves as a storage of energy in the muscle. It's very important for the muscle. This is the reaction. Arginine first combined with glycine by the enzyme amidinotransferase to produce ornithine and guanidino methyltransferase. From this carrier of methyl group S-adenosyl methionine, it is converted into S-adenosyl homocysteine, and the methyl group attached to the guanidino acetate, converting it into creatine. Can be converted into creatine phosphate, the very high energy compound, the energy store on the muscle. And of course, when we need energy, creatine phosphate, it hydrolyzes into creatine, producing ATP. And this is the important store of ATP. Why we don't store ATP directly? Why we convert it into CPK? Because ATP is an important upregulator and downregulator of a lot of enzymes. So it is better to store it as a creatine phosphate, which is not upregulator or downregulator of any enzyme. The availability of creatine phosphate allows the rapid conversion of ADP into ATP and utilization the latter for muscle contraction. We have around 0.5 micromole per gram, for 40 micromole per gram, or 0.5% the concentration of creatine inside the muscle. So creatine is not very stable. Creatine and phosphocreatine, both of them can non-enzymatically hydrolyze to produce creatinine. So this is important. What's the difference between creatinine and creatine? Creatine is very important compound of high energy store. Creatinine have no function. It is the end product of creatine hydrolysis, non-enzymatic hydrolysis, and creatinine is excreted in the blood to be excreted in urine. So it is important for renal function test. It have no importance for muscle contraction, while creatine phosphate are high energy intermediates, important for creatine, as we said, diffuse in the blood, excreted in the urine, and this is very important. How creatine synthesis is regulated? By inhibition of synthesis of a guanidino acetate by creatine. So this is a product inhibition. Creatine inhibits the first reaction in creatine synthesis, which is formation of guanidino acetate by combination of